Sampson was also working in the lab of Michael Summers at the HHMI. And his title is The Structure of the Antiviral Assembly Inhibitor CAP1 Complex with the HIV-CA Protein.
Thank you. Thank you. Again, I'm Sampson Kyere, and this is work I conducted in Dr. Summers' lab. I'll be presenting our structure of an assembly inhibitor bound to the HIV-1 capsid protein. Here's a current look at the HIV pandemic. Currently, there's over 45 million people living with AIDS. In 2006 alone, 4.5 million were newly infected. Regions such as sub-Saharan Africa and Southeast Asia have been hit particularly hard by this pandemic. Current HIV medications target specific stages of the viral life cycle, as I'll show on the next slide. The majority of them target the viral protease or the viral reverse transcriptase. However, due to the rapid mutational rate of the virus, resistant strains are beginning to emerge, making new drug targets necessary. So here's a view of the HIV-1 life cycle. I'll just discuss the late phase where assembly occurs. Assembly is mediated by the GAG polyprotein, which consists of the matrix in green, MA, capsid, CA, and nucleocapsid domains. During assembly, thousands of copies of the GAG polyprotein are synthesized using host cell machinery and co-translationally myristylated, in which a 14-carbon fatty acid is covalently linked to the N-terminus of the matrix domain. After a process of genome recognition, in which the nucleocapsid binds 2 copies of the RNA genome, the GAG polyprotein is targeted to the membrane for assembly. Through unknown, uncharacterized GAG-GAG interactions, you have formation of an immature particle, which is subsequently cleaved by the HIV protease in a process called maturation. The protease cleaves between the matrix capsid junctions and also between the capsid and nucleocapsid, releasing these as mature peptides. The matrix remains associated with the viral envelope in green. The capsid condenses to form its characteristic conical core, which is critical for viral infectivity. And the NC remains associated with the RNA genome. So targeting capsid has been an effective approach in other viral systems, such as hepatitis B, as well as strains of the coronavirus. Within retroviral GAG capsid proteins, there's 2 highly conserved domains. The beta hairpin, which forms subsequent to cleavage by the HIV protease. It initially is a flexible linker and it folds to form this beta hairpin. That beta hairpin is critical for viral infectivity. Any mutations that stabilize or destabilize cause a drastic reduction in viral infectivity. Also, this is the capsid N-terminal domain. Also within the capsid, there's a smaller C-terminal domain. And within the C-terminal domain, there's what's called the major homology region. The major homology region is a stretch of about 20 residues, which is highly conserved amongst all retroviral GAG proteins. It's the most highly conserved sequence in all of GAG. So in our initial aim to find compounds that would inhibit the beta hairpin formation, we came across a compound that bound to another site on the capsid protein, and we previously reported this. In our representative NMR titration spectra, you see a subset of signals shift with increasing concentration of CAP1 during the titration experiments, indicating site-specific binding. As well as we were able to determine that it binds in a 1-to-1 ratio. And we're also able to determine that the KD for this binding is 800 micromolar. In green illustrates the residues that are affected by binding. And this is far from the beta hairpin cleft where we initially tried to inhibit. In our in vivo studies, we were able to determine that CAP1 is non-toxic to the cells, as well as it doesn't affect viral production, but it does cause a 95% reduction in viral infectivity. As well as the reverse transcriptase levels were relative to untreated cells was at the same level as well as capsid production, indicating that it doesn't affect the production of the actual virus, it just affects its infectivity after formation. Here are EM images of the virions produced by these cells. In untreated cells, you can see the central core formation and the majority of the virions produced. However, and also a more homogeneous size distribution of the particles produced. Relative to treated cells, you see a more heterogeneous size distribution, as well as the absence of a electron-dense core within these virions, indicating abnormal GAG-GAG interactions, as well as abnormal capsid interactions. So just to summarize briefly, CAP1 was non-toxic. It showed a dose-dependent reduction in viral infectivity, and it didn't affect any phase of the replication cycle besides the subsequent infectivity of the virions produced. And the EM images showed abnormal morphology, and this activity is different from any other HIV medication currently on the market. So we wanted to solve the structure in order to determine the basis of inhibition. And we were initially able to determine the intermolecular orientation of CAP1 within the complex. CAP1 has a rotatable bond between C7 and M1. And we were able to determine through our nosy spectra here that capsid preferentially binds to one conformation of CAP1. We're also able to determine the intermolecular contacts between CAP1 and the capsid protein using this nosy data here, nosy titration data here. You can see methyls of CAP1 showing NOEs to valines and other residues within the hydrophobic pocket. However, we were not able to unambiguously determine the conformational changes that occur with capsid upon CAP1 binding. In a parallel effort to solve this structure using crystallography in another lab, they were able to resolve great density for the capsid protein, and they saw the structure at 1.5 angstroms, but the CAP1 was not readily resolved in these electron density maps. And this was also due to poor solubility and diffusion from the binding pocket. However, they observed that CAP1 induced a large conformational change in capsid, consistent with our initial NMR titration experiments. So what they observed is that the large conformational change they observed is that the phenyl group of phenylalanine 32 flips from a buried state to a highly solvent exposed state. In this illustration here, the darker shaded helices and loops represent the conformational changes that occur in capsid upon ligand binding, CAP1 binding. The lighter shaded helices and loops are the native state of capsid. So in this closer image of the pocket here, you see the phenyl side chain of phenylalanine 32 flipping from a buried state to a highly exposed state, and this is a 6 angstrom chemical shift or displacement. In our joint refinement, we observed that the phenyl moiety of CAP1 occupies the pocket evacuated by the phenylalanine 32. So we used both our NMR restraints and the structure of the capsid in its bound state in order to do this joint refinement technique. A closer image of the binding pocket shows the numerous hydrophobic interactions between the phenol moiety of Cap1 and the residues within the binding pocket. Also, you can note that the urea group of Cap1 is poised to form a intermolecular hydrogen bond with the valine 59 carbonyl group, as well as outside of the pocket, where the Cap1 is very highly solvent exposed, you see potential ionic interaction between the dimethylamino group of Cap1 as well as to the carboxylate groups of glutamate 28 and 29. So this extrusion of a Phe32 side chain is highly unusual. Review of crystal structures of known capsid, solved of capsid, show that the phenol moiety, the phenol group of Phe32 is predominantly buried. However, there's some unfavorable bond angles observed between alanine 31 and Phe32. And unfavorable bond angles are usually associated with strain in the backbone, and strain in the backbone is usually associated with active site residues. Exposure of this Phe32 relieves the main chain strain observed when Cap1 is bound. So potential role of Phe32 in assembly is that Phe32 is highly conserved across HIV1, HIV2, and SIV, or conservatively mutated. So we speculate that the exposure of Phe32 may be important during assembly. However, the basis of inhibition is still uncertain. Here's a model of the capsid N-terminal domain hexamer that occurs during core formation. And then if you can see, making this out here, these are stick models of Cap1 bound to the apical site of the capsid N-terminal domain. Now this location does not affect the hexameric, will not affect the hexameric interface. However, there's been proposed to be capsid C-terminal domain, capsid N-terminal domain interactions that occur during assembly. But because the structure of the capsid during assembly and the immature state has not been readily resolved, it's impossible for us to know right now the exact basis of inhibition. So conclusions, we identify Cap1 as a potential inhibitor of viral assembly. We use joint NMR and X-ray crystallography to solve the structure of the complex. Cap1 induces a large conformational change in the phenylalanine 32, exposing a deep hydrophobic pocket for the phenol moiety of Cap1 to bind. And we are planning to optimize binding and reduce the KD based on this structure. I'd like to acknowledge Dr. Mike Summers, Dr. Chun Tang, who was the lead author on the discovery of the Cap1, as well as Meyerhoff undergrads, Isaac Kende, Sharika Sylvester, Nurg, Sherry Mofer, and Layla Najimalu. Also HHMI lab members and personnel, and our collaborators that worked on crystal structure as well. Thank you. There's one right there.
This is totally on a different thing, but do you have any idea if there's nucleic acid in your immature particles that are coming off your treated cells?
If there's nucleic acid, RNA?
Yeah.
Yes.
Okay.
RNA is still present. The reverse transcriptase is still active.
Okay.
Yeah.
So you mentioned at the end that you're optimizing the binding. How much better do you have to make the binding before it can be clinically
Okay.
applicable?
Well, current pharmaceutical agents are bind in the nanomolar range. Right now, we're in the micromolar, 800 micromolar, so sub-millimolar range. So within the pocket, there's a lot of potential interactions we can further develop, as well as the Cap1 only occupies about 50% of the pocket. So it's not a full lock and key binding mechanism. So if we add some functional groups to Cap1, change some functional groups, we can further increase bonding.
So will you have to improve it by 10 to the 4th or more?
Excuse me?
By 10 to the 4th?
Yeah, yes. Okay. But right now, we're able to show that see if I have here. Yeah. So right now, intraviral GAG concentration is about 14 millimolar. Based on KD and the passive incorporation of CAP1 into the virus, we can assume that 94% of the intraviral GAG, or the intraviral CAP1 is bound to GAG. So this corresponds to just about 30 or so GAG molecules bound, which would reduce viral infectivity by 95%. So there's not a lot of GAG interactions, or GAG binding that needs to occur to, in fact, reduce infectivity.
One more.
Yeah.
Is it known if the hexamer core still forms in the presence of the CAP1 as the stoichiometry changed, or is that just something that's, that model shows that it's something that's speculated based off of the absence of CAP1 you get a hexamer?
Well, they don't know whether the hexamer still forms in the presence of CAP1. That's actually a model of the MLV capsid protein arrangement substituted by the capsid, by the HIV capsid.